she was making. But then we talked, I don't know, for 3 or 4 hours after her talk. This is about 10 years ago. And we've never stopped talking. Anyway, I'm going to talk to you today about sex differences in cancer. And I am going to unabashedly say at the start, after listening for the past day and a half, that my goal would be to hopefully have somebody think about modeling sex differences in cancer. Because as you're going to see, in particular, growth regulation at the cellular and the body size level, no surprise, it's very, very different in male and female cells, male and female organisms. Cancer risks are different in humans. I'm open to discussion about in animals, as we were discussing before, in non-human animals. And I'd love to see that incorporated into some modeling. And as an aside, I'm an associate editor of the Biology of Sex Differences, the journal for the Society for the Study of Sex Differences. And specifically, my focus is on cancer. And I would love if somebody submitted like one of your modeling type projects on sex differences in cancer. It would just be fantastic. Anyway, sex differences in cancer should come as no surprise. And these are the cancer hallmark pathways that have been made famous by Bob Hanahan and Robert Weinberg. And I'm not going to go into details, but these are the things that change in cancer in order for cancers to grow. And every one of these pathways differs under normal circumstances in males and females. And I'll tell you right now, at the cellular level and at the systems level, I'll tell you like right now that transformation going from normal to malignant cell is not what induces sex differences in cancer. Sex differences in biology exist from the moment of fertilization. What you see is them expressed and interacting with the pathways that drive cancer in the same way that you see them expressed interacting with the mechanisms that drive cardiovascular disease, behaviors as well, mental health, rheumatoid diseases. It's just pervasive, which is why Kristen says that when she looked, she could never look away. Anyway, to start. So a couple of nomenclature things. When I say sex, I am really talking about biological phenotype and nothing else. Yesterday, Melissa talked about sex determination. That's a genetic event, XX versus XY. But we're going to be talking a lot about sexual differentiation, which is primarily an in utero set of events that are the result of those genetics and then a lot of epigenetics. Under normal circumstances, humans are born looking like males or females under normal circumstances. And that's a consequence of events in utero. It doesn't require circulating sex hormone action. And one of the messages here is that sex differences in cancer, with rare exception, are not the result of circulating sex hormones. Differences in drug metabolism can be an effect of circulating sex hormones, but the intrinsic biology of cancer as it relates to the biology of sex differences is not created by transformation. It's established by chromosomal effects, and as Melissa told you yesterday, primarily the non-pseudoautosomal region of the Y, and the process of X inactivation, partial X inactivation, and the clear escape from X inactivation that happened to a whole bunch of alleles that are really important in cancer, including innate immunity, P53 pathway, epigenetic mechanisms. I won't talk about sex-specific reprogramming of imprinted loci, but the sex differences in growth regulation are actually established through imprinting even before fertilization. And then obviously at puberty, there is the effect of activational effects of circulating sex hormones. None of this is gender. Gender, I think for the easiest way to think about it, you should think about behaviors and cancer risk behaviors and how they may differ between men and women as a function of societal norms. For both gender and for sex, there is actually a spectrum of phenotypes. you can go from a maximal masculinizing effect in utero through having an X and Y chromosome and having maximal response to the epigenetic effects of in utero testosterone, or you can have a simple word, a maximum feminization effect of having XX and estrogen. And the power of this to determine phenotype is established by several things. There's a human condition known as androgen insensitivity syndrome, in which there are normal testes formed, there are normal amounts of testosterone produced, and there's no response to that testosterone. And so individuals are born like phenotypically normal looking females. That big phenotypic difference that we see is primarily an epigenetic phenomenon for in utero sex hormones, in addition to the genetic effects. Okay, so what we're going to cover are sex differences in cancer, epidemiology, clinical experience. I'm going to touch on the evolutionary basis for this because it's really important, the developmental origins of it. And then I'm going to give you some examples from our work about sex differences in growth regulation, cellular growth regulation, particularly as it pertains to cancer. And then I'm going to talk about the big challenge in trying to translate the biology of sex differences into clinical care, and an approach we've taken using a Bayesian approach to establish very specific identities for each individual patient with regard to their whole transcriptomes and sex bias natures of gene expression. You'll see that. So females get less cancer than males in humans. This is true all over the world. On the left is cancer around the globe from recent World Health Organization numbers. On the right, cancer in the US. And what's important to take away from this slide is it doesn't matter what genetic ancestry you are. It does not matter what region of the world you're living in. It doesn't matter what your age is, because all of this is true in pediatrics, though in a much more complicated way. Females get less cancer than males do. Females also survive with cancer longer than males do. Everything to the right of the dotted line is an increased hazard ratio for death in males. Everything to the left is an increased hazard ratio for death in females. You'll notice that breast cancer is included in the male side because males with breast cancer almost uniformly die, which is not the case anymore for women with breast cancer. It's really interesting to me that Kaposi sarcoma, which in my experience is primarily an HIV-associated cancer. It's interesting that it's has a higher hazard ratio for death in females. Survival in boys is also shorter than in females. And the importance of this is to remember that there are no circulating sex hormones between the time of birth and puberty. And many of these cancers are happening in that circumstance. So there's no circulating sex hormone effect. Am I making this thing go like this? I'm figuring it is me. Look at that. And importantly, if you look at the graph on the right, which is large and color coded, there's a paper from the last decade. It's from NYU. It's from all their kids with glioblastoma, high grade malignant astrocytoma. They all were treated in that one institution. They were all treated at around the same time period. They all got maximal surgical resection. They all got postoperative radiation therapy. They all got temozolomide chemotherapy. The difference in survival, the females are the green, the red are the males. That difference is just a function of sex. Has nothing to do with access to medical care, doesn't have anything to do with treatment differences, is not related to different stages of the disease at diagnosis. It's sex. That difference in survival is bigger than any single intervention besides surgery with regard to high-grade gliomas, just sex. So it shouldn't surprise us. And I want to start first with development, and I do wish I had a pointer. So early development, pre-implantation development. Most of these data were first generated, I was just saying this, in the meat industry that do everything by in vitro fertilization. Yeah, it's okay, thanks though. In vitro fertilization. And they look for viable embryos through high throughput screening based on metabolism and cell number. And what they discovered across the meat industry species was that from the moment of fertilization, male pre-implantation blastocysts are metabolizing more carbohydrate. They are generating more lactate. They have increased expression of the prime DNA methyltransferases, DNMT3A and B. Their methylomes differ very rapidly. And more recently, their histone modifications differ very rapidly, male and female. And by day 2, male embryos are larger and they contain more cells than female embryos do. These data were reproduced in humans when human in vitro fertilization and the same need to screen for viability in embryos came to be. On the bottom left, you can see basically the mechanisms that are involved pre-implantation. There's imprinting, there's sex chromosome differences, X inactivation, epigenetics, metabolism. There's a brief period of hormonal surge right at birth. And then there's nothing during puberty. Then you see puberty and you see menopause. On the right is a UMAP of male and female pre-implantation embryos based on their gene expression under a variety of different culture conditions. You can see the effect of culture conditions, but the only thing you really need to take away is that all of the females are contained within that red dotted line and all of the males in that blue dotted line, and they are well separated in gene expression space. And we know that this is a profound difference, right? We use different growth charts for boys and girls. They grow at different rates. They achieve their adult plateau height and size at different times and at different plateaus. Growth is fundamentally differently regulated in males and females, and that's been selected for. So Darwin wrote about sexual selection, and I love this quote because he focused on what he could observe, right? So when males and females have the same general habits, but differ in structure, color, and ornament, that's been caused by sexual selection. And so throughout the animal kingdom, we see body size differences, we see ornamentation differences. And the important thing to remember about these ornamentations, like that peacock tail, is they're fantastic for influencing female mate choice, but they are awful for the survival of that individual. They can't run, they can't fly, they can't do anything. The same is true for those big antlers. Fantastic for competition with other males, but really decrease the survival of the individual because the inability to get away from predators in North America, primarily big cats. So there's a cost in individual survival for creating these differences in body size, these differences in color and ornamentation. And why would that be so? It comes down to very simple math, actually. If we think across species, and rather than saying male and female, because as Melissa pointed out, sometimes that can be complicated when you're looking across species. So we can talk about big gamete producers and little gamete producers. For humans, that's sperm and egg. And we can think about per progeny resource sacrifice. So males are capable of making, I couldn't even imagine how many sperm over the course of their adult life. They're physically capable of impregnating multiple females on any given day of their adult life, and just physically capable. They expend no personal resources on the gestation of fetuses, no personal resources on the nurturing of newborns across almost all species. Their per progeny resource sacrifice is as close to nothing as it could be. And so they are said to be mate limited in their reproductive success. And that requires that they have, that big body size, the color, the ornamentation, to influence female mate choice and to intimidate males and compete with males. Females, or big gamete producers, are born with all the ova they'll ever have. 1 to a few become available for fertilization at any given time. Females expend all the personal resources on the gestation of fetuses and the nurturing of newborns. Their per progeny resource sacrifice is actually quite enormous. And they're said to be resource limited in their reproductive success. And so what you see in females is traits being selected for that actually can limit growth, because growth is an enormous resource requirement, and can preserve viability and, as it turns out, longevity. Very different sets of genes that evolution is working on in order for the species, our species and all these other species, to be maximally successful in the generation of newborns. And so Crespi and Summers, 2 evolutionary biologists who thought a lot about sexual selection, proposed that there are 2 different optimums, a female optimum in growth and a male optimum in growth, and that those are maintained by male and female biased actions that push individuals to those optima. And obviously, here's a number of pictures of adult males and females to just emphasize the body size difference. If you look at orangutans, the lower left-hand corner, males are 4 times the size of females normally. That's like a 6-foot male human having a 1 1/4 foot female partner, or a 6-foot female having a 24-foot male. The body size difference is enormous, and it's directly related to cancer risk, I'm going to argue. So size does matter in cancer. And here is the distribution of human heights. The means of these 2 relatively normal distributions is significantly different. The extremes are different. The extremes of normal human height is exclusively male. The extremes of normal female height are exclusively female, but much of the population overlaps in the middle. And we're going to come back to this. It's really important to think about this when we think about sex differences. And here's some data from just a few years back about the way that height relates to cancer risk in humans across cancer types that are shared, and I think some that are not shared between males and females. I can't remember the list now. But you can see the males on the bottom have this very shallow relationship between increases in height and cancer risks. But females have a pretty steep relationship. I'm going to suggest that that's because males are already taller. They're already maxing out on their size as it relates as a cancer risk. And females are really heavily influenced by size. As they gain size that puts them more in the male range, their cancer risk goes up. So based on a day in clinic when all I saw were boys with brain tumors, I'm a neuro-oncologist, pediatric neuro-oncologist, I started wondering about sex differences in cancer risk. And in particular, the way I took the epidemiological data, my personal experience, was to formulate a question was, is it easier to transform a normal astrocyte, because I was interested in glioblastoma, from a male than it is from a female? So Tao Sun, who is a great postdoc quite a number of years ago and is now at City of Hope, did that experiment. Started knocking out tumor suppressors, adding oncogenes one at a time in combination, and found, yes, it was easier to take a normal male cell and turn it into a tumor cell than it was to take a normal female cell and turn it into a tumor cell. And Tao's story focused a lot on the regulation of RB. RB, as you know, is one of the 2 really master tumor suppressors, P53RB. RB regulates cell cycle progression from the G0, G1, which we were hearing about earlier today, into S phase. And you can study RB dynamics by taking away all growth factors in culture. Cells will undergo arrest in G0, G1, and then you can add growth factors back. And you can watch for the hyperphosphorylation of RB, which inactivates it and leads to its degradation. And you can watch the onset of cell cycle progression again. Males are on the left, females on the right. We're Western blotting for phosphorylated RB, total RB, actin as loading controls in those two parallel gels, and you have no trouble seeing, I think, that RB is much more rapidly phosphorylated in male cells, and the magnitude of that phosphorylation event is very much higher. And their proliferation rates, which are up in the upper left-hand corner, for cells that have lost neurofibromin, which is a tumor suppressor by suppressing the activation of the MAP kinase pathway, and P53, which is the master tumor suppressor in terms of size and cancer risk. Peto's paradox, elephants don't get cancer. They have 48 P53 alleles. We have 2, and it's just what Peto predicted. And if we implant those cells that have loss of neurofibromin and loss of P53 into the brains of male and female mice, doesn't matter what the sex of the host was. It matters what the sex of the implanted cells were. All of the recipient animals, male or female, died from male cell implantations. Only 30% died from female cell implantations. Cells were labeled with green fluorescent protein, so we could find them. They didn't die because of implantation. They did not form tumors. And you'll notice that there appears to be at least 2 groups in the females, some that rapidly died and some that didn't. And this brings me to a second point about the importance of thinking about in utero sex hormones and patterning of gene expression and phenotype. In animals that give birth in litters with single placentas, female pups are partially masculinized as a function of the total number of male pups, so the proportion of male to female, and the position of the female pups between male and female. So we do all of our experiments with at least 5 astrocytes derived from at least 5 different litters of mice, because we need to capture that range of sex differences. That's what happens in humans through different mechanisms, big range in sex differences. We recapitulate that by using multiple different litters that have different compositions. And so those animals that died as fast as males, not surprisingly, came from litters that had mostly female pups. I mean, mostly male pups. They were presumably partially masculinized, while the others did not. And that's why we do the experiments that way. I'm going to very quickly talk about this growth regulatory mechanism, because I want to highlight some of the ways that we think about analyzing sex differences. So this was work done by really amazing MD PhD student, Lauren Bristol, who's now off to UCSF for a neurology residency. So Lauren was looking at, wait a minute. I mistakenly deleted the one slide that included work I'd done with Kristen. Maybe it wasn't all by mistake. No, it was absolutely by mistake, because it was the lead into this. Using Kristen's algorithm for calculating tumor growth velocity, we looked at human GBM patients. And what we determined was that with after radiation therapy, repeated cycles of temozolomide, chemotherapeutic, were associated with progressive decreases in female tumor growth velocity, but not in males. They had an initial decrease and then it plateaued. So again, I would love to talk to somebody about modeling this response. So we were interested in understanding the mechanism. And so Lauren was studying responses to irradiation in our model system, and found that we could see the exact same thing across 5 litters of response to radiation. females were more sensitive to it. And remember, when we talk about survival data after cancer, we're really talking about response to treatment, right? We're not looking at survival in individuals who aren't treated. We're looking at treatment response and treatment resistance, as you guys have been talking about a lot. But what we find, because of the range of sex differences, is we can't treat sex as a categorical variable. It's not a categorical variable. It's a continuous variable because of the way that it exists as a spectrum of phenotypic differences. So Lauren was very focused on this regulator of cell cycle arrest in response to DNA damage, P21. It's a transcriptional target of P53. It is required for the growth arrest that happens after radiation and DNA damage in chemotherapy. And it is also the mediator, depending upon the amplitude of its response and the kinetics of its response, for promoting cellular senescence, which is a therapeutic response to radiation and chemotherapy initially. It can then be a tumor promoting response because senescent cells secrete a lot of growth factors. But so Lauren was correlating a measure of cellular senescence, senescence associated beta gal positivity, with the relationship between gene and protein expression for P21 and its target, the cyclin-dependent kinase 2. And you can look at the Western blots above. The point of this is that the way we study this is by doing regression analyses. We can't compare males to females and evaluate it with a t-test. We have to look at the correlations and evaluate the slope differences. And so what I'd like you to see is in the 24-hour, 5-day graphs on the right below the Western blot are the protein expression of P21 and CDK2 as it relates at 24 hours and 5 days to senescence after irradiation. And you can see that the females have a much steeper sensitivity, greater sensitivity to the induction of P21 than the male cells do. This is hugely important. This is why they respond better. When we delete P21, I'm not going to show you that, we lose the sex differences. Females respond like the males, like those rapidly dividing female implants. But you got to do it by regression analysis. Oh, see, here it is, but it's in the totally wrong place. On the left is Kristen's data from GBM specimens, and on the right is Lauren's data with regard to irradiation. In between, we can forget for the time being. But if we look at just decreases in cell number in response to radiation, if we look at regrowth after irradiation, if we look at colony formation after radiation, we see the same thing. Females are more sensitive to radiation and they have less regrowth after radiation than male cells do. Anyway, out of order. In sex differences world, you can ask about the contribution of chromosomal and gonadal sex to any aspect of biology. For us, we're using postnatal day 1 astrocytes, so we're really interested in utero development. And so the way we do this is by using something known as 4 core genotypes model of sexual differentiation and sex effects. It was developed by Art Arnold, who really is, like, along with Margaret McCarthy, the pioneers of this field. And the way this works is deleting that single gene from the Y chromosome that's necessary and sufficient for the formation of testes, SRY, and cloning it onto an autosome. So now you can get 4 different genotypes in a litter. You can get, it's pictured in those boxes underneath the circle and square above. You can get XY individuals, chromosomally male, that have SRY, so they're gonadally male. You can get individuals that are XY, chromosomally male, but SRY negative, so they're gonadally female, and the same complement with the XX. And this allows you to ask whether or not the sex differences you're seeing in biology are patterned by chromosomal or gonadal effects or a combination of the two. And the way that you look at this data is pictured on the right. Again, I'm just trying to illustrate how we do these experiments, how we think about these data, how we come to our conclusions. If you look on the left, we're looking purely at the chromosomal effect. We're comparing any mouse that's XY to any Gesundheit, any mouse that's XX, and we're looking at that same relationship I was showing you before, but now we're looking at it at the mRNA level only. And you can see it doesn't matter what your chromosomes are. There's really no correlation, chromosome sex and senescence and CDKN1A, which is P21, and CDK2. But if you look at the SRY status, what you see is that having the absence of SRY, being gonadally female, is what determines that response that we saw in females. SRY positive individuals don't show this relationship. SRY positive individuals, those that are gonadally male, are the ones that didn't respond to radiation therapy as well. They're the ones that didn't undergo radiation-induced senescence. They're the ones that Tao found rapidly reestablish cell division after arrest. It's patterned by in utero gonadal hormones through epigenetics. These things matter a lot in the clinic, and if we looked more, I'm certain they would matter more. Here's just some data analysis of current clinical trials and past clinical trials that used MEK and BRAF inhibitors, primarily in melanoma. And you can see that the males and females actually have quite different responses to that. In this case, females are responding better to treatment, as is true in general. That's why the red data points are to the left of the blue data points, which are the males. So it's important to be thinking about this. And most importantly, it's important to think about this proactively, prospectively, not looking for retrospective fits to data, but powering clinical studies, laboratory studies to detect the sex difference. So, but here's the challenge when we think about bringing this to the clinic. So I don't know how many of you recognize this fish, but the big fish there is an adult female anglerfish. The two little fish are two adult male anglerfish. When those adult males get to sexual maturity, they begin to auto-digest their mouth to anus. They search out the perineum of a female. When they find one, they secrete enzymes to degrade her skin, and they secrete angiogenic factors to promote the ingrowth of blood vessels. When their circulatory systems are hooked up, they auto-digest basically all of their internal organs, and they spend the rest of their lives as testicles and nothing more. This is sexual dimorphism. You'll hear this term misused all the time. We as humans are not that sexually dimorphic. In fact, we're the least sexually dimorphic of primates. What we see clinically and in humans is what's captured by the height comparison. We don't see dimorphism. What we see is two different normal distributions with two very different extremes and a lot of overlap in the middle as a consequence of male and female biased actions regulating growth. This is the clinical problem. So you can't be thinking about a male specific treatment versus a female specific treatment. That's never gonna work, except if you're treating individuals at those extremes, but most patients are not at that extreme. So how do we deal with this? Well, I want to give you some real data to illustrate what you can see. I'm not just making this up. So this is the work of a former graduate student who's now a postdoc, not in my lab, who was looking at metabolite abundance in male and female glioblastoma patients. So this is a metabolite abundance heat map. I mentioned before that metabolism differs from the moment of fertilization. Female cell metabolism is focused on fatty acid metabolism. Male cell metabolism is focused on carbohydrate metabolism. They have very different amino acid metabolisms. And what's captured here is metabolite abundance. And this difference, I'm going to point out the difference in a second, is very similar to what you find in normal, healthy adults when you look at metabolite abundance in the serum Again, not created by transformation. Yellow is most abundant, purple is least abundant for these metabolites. And what you can see on the left, the bar on the top is indicating in blue and red what's male and what's female. The standard in the field is blue is male and red is female. So you can see that metabolite abundance is skewed, right? The higher levels of metabolite abundance are towards the males. And in fact, there is a clear extreme there, that's all males. And then the lower metabolite abundance is where the females are. But there's a whole overlap in the middle, right? Real world. And this matters. So on the left here are images from patients treated at Sloan Kettering. They're standard anatomic MRs, and there is glutamine PET imaging. Among the metabolites that Yasmin found in that metabolomics study that are very different between males and female tumors and also different in serum are everything in the glutamine, glutamate to both glutathione and then to TCA cycle metabolites. Glutamine deficiency targeting the generation of glutamate from glutamine with antagonists of GLS are currently in clinical trial. What you can see on the left with the PET imaging for glutamine in this single illustrative case, but the cumulative data are on the right, is that the male tumors tend to uptake more glutamine than the female tumors do for the same sized tumor, relatively, same enhancing qualities. On the right is comparison of the glioma to the normal brain. And you can see that the males have a statistically higher distribution of glutamine uptake than the females do, as does the normal brain. And once again, you can see that at the extremes, the highest points, all male, extremes, the lowest points, all female, most everything is overlapping in the middle. We can quantify the glutamine consumption in our cultures, and you can see, and we chose a single litter for this study that had maximum male-female difference because we just wanted to illustrate how important sex differences can be. So I just want to be clear about that. So here we're using the t-test and we're just looking at the sex difference in glutamine consumption. Males consume more. On the right, we're looking at the sensitivity to glutamine deprivation. You can see that the females are less sensitive to glutamine deprivation than the males are. And on the very right is response to the clinically available and currently evaluated GLS inhibitor, CB839. And you can see that the males are quite sensitive to it and that the females are not. This will be important in the clinical trials that are currently being done, except the clinical trials are not at all designed to detect sex differences. So the consequences of this will only be approachable retrospectively and will not have the power, I can guarantee that, to detect this. So what do we do about this? What do we do about patients in the middle? So going to go back to that kind of distribution of heights, but now I'm going to talk about glutamate abundance or glutamine requirements. Just as a way of entree into talking about this approach. And I'm going to remind you of male and female biased actions. How do I know how much time I have? But it's counting up. It says that I currently have 22 minutes and 10 seconds. Now I have 22 minutes and 15 seconds. So somehow or other, and I know I've been talking for more than 22 minutes. Thank you. Oh, I have a call. It's 11:40. I'm supposed to end at noon? Perfect. No? When? Yeah, can I talk a couple of minutes longer? I'm sorry. I'll try and go fast. What? Okay, I'll try not to be too long though. So what we want to be able to do is individually determine the ratio of male biased, and I'm going to suggest we need to move away from the sex-associated terms. So let's talk about the high bias and the low bias. Everybody's got a mix of high and low biased mechanisms, except for the people at the extremes. So how are we going to deal with them? We want to identify a single identifier that would allow us to do some treatment planning based on patient sex. So the way we did this is we took whole transcriptomes from close to 9,000 patients from TCGA and then a couple of pediatric data set sources. And we clustered them based on the similarity in their transcriptomes. And in two-dimensional space, what you see, I'm going to show you in a second, is that you'll find local clusters of male and female cases. And using those local clusters as what we refer to as pole-defining transcriptomes, and there can be multiple poles for each sex within a large data set, we can now calculate a Bayesian distance based on posterior probability of any individual being identified as male and female just based on their transcriptome. And we can set the axis, the x-axis up now, not as height, but we can set it up as a distance of 0, which we refer to as a transcriptomic sex index, or TSI, which you'll see on the graphs. It starts at 0, which by similarity to the height and the female distributions, that's the female pole, and a distance of 1, or probability of 1, and that's the male pole. And this is what we get. Oh, I just said that. This is pan-cancer ridge plots for TSI, or Bayesian nearest neighbor distances on the left. You can see that just like height, male and female distributions are distinct. There's a male pole, there's a female pole, and there's a lot of overlap in the middle. If you put them all together, it looks like one normal distribution, but that's not an accurate representation of the biology. If we look at the genes and pathways that distinguish the poles across, this is for adult cancer, so down in the bottom, or 29, I think, adult cancers. And we look at the genes and the pathways that are associated with the poles. There's no single one pattern, but the dominant patterns are these. If you look at that block of red circles, the intensity of it is the numbers, the size of the circle is the number of genes involved. The intensity of it relates to the FDR rate. The more intense, the lower the FDR. So there is that big red block there across multiple cancers in females. It's all immunity and inflammation pathways. And it's well known that male and female immune systems function very differently. The other big blocks are the blues that you can see down in the lower left corner, and that's all cell cycle regulation. And it's known that cell cycle regulation is different in males and females. Also in males is oxidative phosphorylation. And then sarcomas are interestingly flipped. They're the ones all the way over on the left. And that's true in adults and pediatrics. I have no idea what that relates to. Pediatric genes and pathways are very much the same. The ridge plots look very much the same. So what can we do? So if we look at clear cell carcinoma of the kidney, trying not to be totally GBM focused, we can see that the female pole is defined by all of that immunity inflammation signaling, and the male pole is defined by epithelial mesenchymal transition, oxidative phosphorylation, and late estrogen response. I want to point out that much of testosterone effects are mediated by the conversion of testosterone to estradiol. In fact, most of the brain masculinizing effects are actually estradiol mediated. So estrogen response pathways are always active in males. So if we were to take two individuals with identical TSI values in the middle, and we looked to see whether or not the same pathways brought them to the middle, and therefore maybe which pathways we should target, what we find is that they're very different. So in one regard, we see downregulation of things that are part of the female pole. So this is females moving to the mid-range. So you see a decrease in some of that inflammatory signaling. Interestingly, you see a decrease in fatty acid metabolism. And as I mentioned before, female cells naturally are fatty acid focused in their energy metabolism and their anabolic, their use in anabolic reactions. But you see a gain in EMT, which was male pole. You see a gain in estrogen response, which could work both ways. You see a gain in response to DNA damage, and you see a gain in an oncogenic pathway, KRAS. So what brought females to the midline is losing a little bit of their mid-range, losing a little bit of their pole-defining pathways and gaining some male pole-defining pathways. What you see in males is decrease of several things you might have predicted: oxphos, glycolysis, they're carbohydrate centered in their cellular metabolism. You interestingly see an increase in mitotic signaling, which is kind of counterintuitive and might relate to some of the things that you guys have been saying about what happens when you're stressing and how I'm going to say non-canonical some of the proliferative responses can be. There's an increase in inflammation and an immunity signature. So if you were dealing with two clear cell carcinoma patients with identical TSI values in that mid-range, one male, one female, you would be thinking at a molecular tumor board, I'm going to target different pathways in these two individuals, even though their transcriptional phenotypes are the same, because the mechanisms underlying how they got there are different. And here's a case illustration, and this is the last data slide. So in lung cancer, it is very well known that females do not respond to immune checkpoint inhibition alone. Males respond quite well to it. And the hypothesis, which is simple enough, is that female anti-tumor immunity at baseline is very high. So using a therapy that's meant to stimulate immune response just really doesn't have much effect. Males have low basal anti-tumor immunity, and so you can stimulate it, and that's what you see. So if we take our transcriptomic signatures and we say that the immunity signature in females is a resistance mechanism to immune checkpoint inhibition, it's poor use of resistance in a way, but it makes sense practically. And that the weak immunity signature is the signature of sensitivity to immune checkpoint inhibition or responsiveness. Let's say responsiveness, non-responsiveness. You can look at the ridge plots, you can overlay the signature and the sensitivity. And if you were to conduct a clinical trial where you wanted to use immune checkpoint inhibition alone as a treatment, and you were required by the NIH or whatever funder you had to recruit equal numbers of males and females, you could choose from these distributions which females would be most likely to respond and which males would be most likely to respond. And that can be a selection criteria for enrollment. We do that kind of selection criteria for lots of targeted therapies. What is your gene expression? You could similarly take everybody in the middle and you would predict responsiveness based on the strength of the anti-immunity signal. So the immunity signal. The male response would be declined compared to its pole, I'm going to predict. Female response would be greater compared to its pole. And so you can construct the x-axis any way you want. We've done experiments having to do not just with cell cycle regulation, cellular metabolism. We've done epigenetic mechanisms. You can always set up an x-axis for a targetable pathway that would allow you to incorporate patient sex as it exists in a spectrum of high and low biased actions in biology for treatment. And with that, I've just said that. Just going to thank my wonderful lab and my wonderful collaborators. Kristen's up there. Sorry, you came after Rosie, because Rosie does everything for me. Anyway, thank you.
Take like one, maybe two questions tops, and then we'll move on to the rest of the session.
I was hoping somebody would challenge me. What's that? All right, well, I welcome, I really love scientific discourse, as you may have noticed. And so I'm hopeful. I need a list of the speakers so I can do. Oh, question. Yeah. I thought you were a speaker.
It's kind of rather practical question. So obviously, so the tumors are not just about what's happening in the tumor cells. There's also the systemic effects and microenvironment. And like working with xenograft models, and it's not uncommon to observe clear differences in responses or engraftment rates between males and females. And again, naive thinking would be, okay, it's probably about the sex hormones, but as you pointed out, it's often like many other things that are working in additional to the sex hormones. So if encountering a phenotype with a difference between male and female host, what would be sort of the advice in terms of trying to figure out what's going on and what to look for?
So fantastic question. So there's several things that I would suggest doing. So one of the more common reasons for differences in engraftment between male and female tumor cell lines, I think it's important to remember that cell lines are actually quite different from freshly isolated cells, has to do with anti-tumor immunity. So if you're using nude mice and you see that, that tells you that it's not an adaptive immune response that's creating the difference. If you've done it with syngeneic implants and you see it in an immune competent mouse, the first thing I would suggest is doing the nude mouse and seeing whether you see the same difference and whether it has any relationship to adaptive immunity. Nude mice still have innate immunity, and much of the anti-tumor effect is an innate immune response. So my first suggestion would be to either manipulate or analyze your xenografts for the infiltration of innate immunity cells, because in the literature, when looked at mechanistically, the sex difference in engraftment, that's frequently the case.
But I mean, actually, some different scenarios. So the cells are identical, but they kind of, the take rate is different.
Oh, in males and females. Right.
So there must be some systemic factors.
Exactly, anti-tumor immunity.
And in that case, it's like NSG mice, so they don't have any adaptive immunity, even
right.
NK cells. So it must be something else.
So if that's still present in the nudes, it's still likely to be anti-tumor immunity, but it's likely to be the innate immune system, not the adaptive immune system, which you can evaluate from tissue sections or you can manipulate. There are plenty of antibody reagents for depleting innate immune cells, which would be a relatively simple experiment. It's almost always immunity, not always, but almost always.
Hi, great talk. Quick question. It has been described that across species, the larger
animals,
the larger individuals survive less than the smaller ones. So, do you think in your effects that size has anything to do with the sex differences you see?
So let me think about the variables here. So you're asking about a variable that has to do with sex and size as it relates to sex, and size as it relates to cancer risk, and size as it relates to longevity. Is that correct?
Yeah.
All right, so 4 variables. So longevity tends to be longer in females than it is in males. And with regard to longevity being a risk factor for cancer, because it's the single greatest risk factor for cancer, despite the fact that females live longer, they get less cancer. So the longevity part, it has a different effect in males and females. With regard to the size component, females tend not to get to very large sizes, even at the tallest of females, they tend to be well within the range of male sizes. And so if it was just a relationship between size and cancer, I think it would be like what we saw, where the males are already tall. There's not that much difference in cancer rate as a function of size in males, but there's big difference in cancer rates in females because they're starting off shorter. So that effect of size on cancer risk is magnified. Does that answer your question? Yeah. Okay.